More than 20 years ago, we discovered polyglutamin-binding proteins with the aim of finding binding partners to causative proteins of polyglutamine diseases. Polyglutamine disease includes 9 neurodegenerative diseases such as Huntington disease, cerebral ataxia, Kennedy's disease, and so on. Their causative proteins possess expanded polyglutamin tract sequences. Therefore, we suspected there should be some physiological proteins binding to the polyglutamin tract sequence and mediating the pathology. PQPP1's function is already known, but the others remain unknown. Nucleolus was first observed by Felicitas Fontana in 1781. Since then, a huge number of knowledge has accumulated, and now nucleolus is considered to contribute to essential biological functions, including cell proliferation, regeneration, cancer, aging, and neurodegeneration. Grossly speaking, the nucleus is mainly involved in messenger RNA transcription, while nucleolus is involved in ribosomal RNA transcription. I'd like to introduce you to a finding that our original molecule PQPP5, which is also called NOR10, plays essential roles in nucleolus. The nucleus has a double-layer membrane and is separated from cytosol. However, nucleolus does not have such a membrane. Therefore, it has been a big question how nucleolus is made in the nuclear matrix. In addition, it is not explained how nucleolar substructures are made, such as fibrillar centers, dense fibrillar components, and granular components. Recently, nucleolus has begun to be considered as an assembly of multiple intrinsically disordered proteins, in brief IDPs, based on a physical phenomenon called liquid-liquid phase separation. In this study, we found PQPP5 is an IDP by high-speed atomic force microscopy and by ultra-resolution microscopy. We revealed that PQPP5 forms a lattice-like meshwork ball to which other nucleolar proteins attach. In the absence of PQPP5, nucleolus could not keep its normal structure or disappear from the nucleus. Interestingly, in some neurodegenerative diseases such as Huntington disease or cerebral ataxia, their disease proteins sequester PQPP5 to their aggregation foci and thereby induce a similar situation to the absence of PQPP5. Consequently, abnormal nuclei are frequently observed in the model mice of Huntington disease or cerebral ataxia. In summary, our study leads to two conclusions. First, PQPP5 is an essential component for nucleolus structure. Second, PQPP5 contributes to neurodegeneration via nucleolus. Previously, we showed PQPP1 interacts with ataxin-1 and huntingtin and mediates Huntington disease and cerebral ataxia. Now, we showed PQPP5 interacts with ataxin-1 and huntingtin. This means that IDPs and disease-causative disordered proteins DDPs will have wider relationships in their functions and the pathologies of neurodegenerative diseases, just like threads and textiles. The full understanding of the world woven by IDPs and DDPs will lead to our comprehensive understanding of neurodegenerative diseases. Thank you for watching.